Yeah, thanks for the kind introduction. So just to say, this is a pure coincidence that the term tree structured appears in the title as well. We work with a different problem. So our work is concerned with learning fitness landscapes from tumor mutation trees. So as we know, cancer progression involves a somatic evolutionary process where cells accumulate selectively advantageous mutations that allow them to evade immune surveillance and proliferate uncontrollably. And as with this increasing availability of single cell DNA sequencing data, we are now able to reconstruct the evolutionary histories of different tumors at very high resolution and represent them using the tumor mutation tree. And here we show an example of such tree, and the nodes in the tree represent the mutational events, and they are ordered according to their occurrence in the evolutionary history. And the cells attached to the same node form a subclone, and their genotype is obtained by accumulating the mutations from the root to the node where the cells are attached. And instead of looking at just one tree for a single patient, we actually sequence tumor samples across different patients and try to analyze a cohort of tumor mutation trees. And these trees are often highly heterogeneous, namely that you rarely see a single evolutionary trajectory appearing more than twice in a cohort. And that makes cancer very hard to treat as well, and highlights the complexity of the underlying evolutionary mechanism. And our goal here is to really disentangle different evolutionary forces that shape the different tumor subpopulations. And more specifically, we try to infer the fitness landscapes of mutations given a set of tumor mutation trees. However, most existing evolutionary models only focus on one aspect of the data. At one end of the spectrum are the graphical models, which focus on inferring common mutational patterns or repeated evolutionary trajectories from the tree topologies only. And these methods ignore the abundances of different subclonal populations and therefore could not be used to quantify fitness. At the other end of the spectrum are population genetics models, such as the well-known Moran process, Wright-Fisher process, branching process. And these models address how different evolutionary forces shape the mutation frequencies over time. So this set of models is perfect for inferring or quantifying fitness. However, they do not exploit the evolutionary relationships encoded in the trees to inform their estimates. And very few methods really lie in the intersection, and those that do are typically only designed for a single patient or single tree. So that means we need a new method that can a new principled framework that can merge these two seemingly orthogonal data, model classes. And we base our model on the branching process, a flexible and intuitive stochastic process for modeling cell proliferation, death, and mutation. And here I'll show a very small example of a two-type branching process model for colorectal cancer. And there's a stem cell compartment where the population size is static. And these normal cells can accumulate mutations stochastically at rate mu1 and turn into adenoma cells in compartment A. Then these adenoma cells can duplicate at rate B1 and die at rate D1. And at rate mu2, they can again turn into malignant cells, which then duplicate at rate B1 and die at rate B2 and die at rate D2. And on the right, we can see the corresponding set of reactions. And already with this very simple system, we can generate highly heterogeneous trajectories of adenoma and malignant cells at different time points. And this level of variance is ideal for modeling the heterogeneous cancer evolutionary process, which cannot be adequately captured by the deterministic models as illustrated by those thick lines, which represent the expected population sizes. Analogously, tumor mutation trees can be interpreted as realizations of a branching process. In this view, the compartments correspond to the subclones, and each compartment size reflects the number of cells attached to those subclones. And it follows that the probability of observing a certain configuration of subpopulation is just equivalent to the probability of observing the corresponding tree topology and cell attachment at time t. Therefore, we could use branching processes to model tumor mutation trees. Now we are ready to formally define fit tree, a tree structure branching process with epistatic fitness effects. Conceptually, this is a birth-death mutation model for generating trees. We assume that the evolution starts with a static wild-type population, and this could be, for example, hematopoietic stem cells in the bone marrow or intestinal stem cells in the colorectal crypts. Then we define a tree expansion rule that determines the possible genotypes or subclones allowed in the tree, also called compartments in the classical branching process model. And these compartments contain 0 cells at the beginning of the evolution. And examples of such rule would be infinite size assumption or relaxations that allow for parallel or back mutations. So in this sense, we are very flexible in how the tree structure can be defined. We then define the growth dynamics for cells within each mutant subclone, similar to the 2-type example. We assume that each cell grows independently of each other, and then a cell can emerge from the parent subclone through mutation at rate nu, and then duplicate at rate alpha and die at rate beta. And this set of reactions applies uniformly to all mutant types. For identifiability, we assume that all cells share the same death rate. And to capture potential epistatic effects, we parameterize the log birth to death ratio using a triangular fitness matrix F. The diagonal elements in the matrix represent the fitness effects of individual mutations, and the off-diagonal elements account for the pairwise interactions between the mutations. And here we use a max sum formulation, which assumes that only the most advantageous mutation in the subclone will dominate its fitness, and all epistatic effects are added through the interaction terms. Next, we define a sampling event whose rate depends on the tumor size. And the underlying principle is that larger tumors are more likely to be sampled. And this makes sense because patients are more prone to exhibit phenotypic symptoms and seek medical attention. Upon diagnosis, a sequencing event follows where a much smaller number of cells are drawn without replacement from the tumor cell population. And after that, we will treat the leaves of the tree with 0 sequence cells as unobserved and prune them from the tree. And this results in the observed tumor mutation tree. Now we have a complete stochastic process for generating trees. We then can, in principle, compute the probability of any given tree. However, it turns out this is technically very challenging. Returning to our 2-type branching process analogy, even in this very simple case, like the exact solution can become very involved and can only be expressed using a very ugly probability generating function with several hypergeometric functions. And so far, for systems with more than 2 cell types, no exact solutions have been derived. So let alone for trees, then how exactly can we compute the probability of a tree? And the key contribution in our work is exactly to approximate those probabilities. By generalizing prior results for simpler linear multi-type branching processes, we show that for any given subclone in the tree, in the limit of large time and small mutation rate, the subclone size distribution CT can be decomposed into a time-dependent deterministic function and a time-independent random variable. Okay, then why does it matter? Because we typically only observe a single snapshot of tumor at diagnosis and cannot access the full trajectories of all subclones. And this result allows us to project their growth trajectories back in time, bundling all stochasticity to the time-independent random variables. And this allows us to compute the tree probability relatively easily. And now it can be decomposed into 4 parts. First is the population densities of the subclones directly following the roots, such as the red node, and they follow negative binomial distribution depending on the net growth rates. And the second term will be all the population densities for all populations, all subclones further downstream. So given the parent sizes, we can compute the conditional probability distribution of their sizes using inverse Laplace transforms. And we have probability distribution for the sampling time event, and this depends on the entire tumor, and this is inhomogeneous Poisson. And lastly, we have the distribution for the sequence cells, and this is since it's drawn without replacement from the tumor cell population, this is a hypergeometric distribution. And with this likelihood function, we are then able to perform Bayesian inference to sample from the posterior distribution of fitness matrix given a set of tumor mutation trees. And we make the following assumptions. Tumor sizes can be estimated from clinical measurements, and the sample proportions accurately reflect the true subclone frequencies. And third, all parameters except the fitness matrix F can be derived from literature or clinical data. In the first part of our simulation studies, we demonstrate that across a range of parameter settings, our method can accurately approximate the densities in the tree likelihood. Here we can see that the theoretical CDFs match almost perfectly with the simulated CDFs from the Gillespie algorithm, and this ensures reliable fitness landscape inference in downstream analysis. In the second part, oops, I don't know why the colors went away, but we show that the fit tree outperforms state-of-the-art methods in learning fitness landscapes from simulated trees in terms of mean absolute error sign, agreement, and Spearman correlation. And the competing methods include a replicator model, a diffusion-based approximation of a one-type branching process, a Wright-Fisher process, and the simple baseline that based on only mutation frequencies. We applied FitTree to a single-cell AML dataset consisting of 123 patients and 15 genes present in at least 3 patients. The inferred fitness matrix reviews a complex interplay of positive and negative epistatic effects. Positive interactions are often corresponding to known co-occurrences such as NPM1 and FLT3. Yeah. And while negative ones may reflect clonal exclusivity between terminal mutations such as FLT3 and NRAS. Finally, we made predictions of the next likely mutational events given a primary tumor tree and validated using longitudinal samples. And here we show one example of such a case, where we can see this mutation WT1 was absent from the diagnosis sample but appears in the first post-treatment sample. We have 4 such cases. We rank the instantaneous growth rates among all possible future events for one given tree. And the higher the percentile is, the more likely the event is going to happen, as indicated by the model. And we see that the fit tree assigns high rankings to those events that actually happen. And it is and the percentile rank is consistent with the consensus estimate from other methods. And additionally, fit tree provides this 95% highest density interval, allowing us to quantify the uncertainty in each prediction. In the end, why do we care so much about defining the tree generating process and estimating the fitness landscapes? Because our goal is to build a principled framework that goes beyond modeling primary tumor trees and ultimately to enable treatment outcome prediction. And FitTree makes this possible because the treatment-induced changes can be incorporated into the fitness parameterization, allowing us to simulate relapse dynamics through forward sampling. We are now extending this framework to integrate clinical covariates, mutation effects, and drug effects. Yeah, so that brings me to the end of my presentation. Many thanks to Nico, Jack, Kevin, and everyone in our computational biology group for their support, and to my collaborator Koichi for kindly providing the data. And this work was supported by SIB and skintegrity.ch, and I'm also honored to be supported by ETH and ISCB to attend the conference. Thank you for attention.
Hi, thank you very much for your talk. And we're just wondering if you talk about prediction patient outcome. So I guess for some of the samples, you already have the patient survival time. How do you envision actually how do you use the tree to actually predict that?
Thank you for your question. This is a very, very nice question. So in this part, yes, indeed, we are now integrating all the longitudinal samples, and we are actually expanding our data sets to have more patients as well. So since this is a branching process model, naturally we can do forward simulation with the model. So we could have naturally the probability of the survival time. So this probability of the sampling time can somehow be extended to model the probability of the survival time. Yeah, thank you.
I think we have to move on. Let's thank our speaker again.